Hi, everybody, and welcome to this week's lecture. What we're going to be looking at today is going to be an introduction to the cell cycle. So our main objective is just to understand and learn the steps of the cell cycle, exactly what goes on with it, what are the different phases, and what are the different checkpoints. So let's go ahead and get started by talking about the cell cycle. Now, the cell cycle, just like the name suggests, is a cycle that cells go through in order to divide, in order to kind of help with growth, repair, things like that. So again, you can see that we have an image over here, and it's showing our cell division in our cell cycles. You can see over here that we're starting off with certain cells. You can see that we have some portions where they're going through different phases, and you can see that its ultimate goal is to go from one cell into two cells. Now, remember, when we're going through cell division, specifically with mitosis, we want to make exactly the same type of cells that we're starting with our original. And we're going to end up with something known as daughter cells, because again, they're coming from the original piece. So again, in our cell cycle, we have a complete cycle that it's going to go through in order to go from this one piece right here into two identical cells. So let's go ahead and check out the exact stages of the cell cycle. When it comes to this, we have two major portions that we need to understand. The first is going to be known as interphase. Now, interphase is going to be the portions from here all the way through here. Now, one thing you'll notice is that interphase is going to take up majority of the cell cycle. Now, in interphase, all that's happening here is the cell is going to grow and make copies of its DNA. It's not until we hit our M phase or our mitotic or mitosis phase is when the cells are going to physically separate its DNA into two sets, and it's going to divide its cytoplasm, forming those two new cells. So again, when it comes to the cell cycle, interphase is where the cell spends majority of its time, and it's not until that very end portion with that M phase is it going to physically separate out, going from that one cell into our two cells. Now, as we're looking at the stages of cell cycle, we have different phases in each one of our pieces. You'll notice that we have a G1, an S, and then a G2, and then ultimately going to our M phase or our mitosis phase. Now, when it comes to our G1 phase, all that's happening here is that the cell is physically growing larger. It's going to copy its organelles, and it's going to make molecular building blocks it needs for later steps. So think about our G1 phase as kind of a getting ready phase. It's getting ready in preparation to kind of go through that cell division. In our S phase, this is going to be called the synthesis phase. So a copy of that DNA in its nucleus, it's also going to duplicate its cytoskeleton structures called the centriomes, and it's going to help, which are going to help separate DNA during the M phase. So in this case, synthesize, think about the S phase as it's making complete copies of that DNA in that nucleus. Okay, so again, now we're getting ready with that DNA in order to have that second set so we can go ahead and go through that cell division, so we can have, again, everything we need and have identical copies when it comes to those daughter cells. So our G2 phase, again, during this gap phase, cells are going to grow more, make more proteins and organelles, and begin to reorganize its content in preparation for mitosis. So in G2, it's really kind of rearranging everything and getting everything settled for that actual cell division portion. So again, G1, it's preparing, it's growing larger, it's making those copies. S phase, synthesis, it's producing or making a complete copy of that DNA. And then in G2, it's really settling down to get ready for cell division. Now, in that M phase, in mitosis, this is where the cell is going to, again, divide out into two pieces. So again, during the M phase, cell division, cells divide their DNA into two identical sets of chromosomes. So again, we're really getting into that portion now to where it's going to divide out. So these are going to be our phases in between our cell cycle. So again, quick recap, we have two sections or major phases when it comes to the cell cycle. We have our interphase where the cell spends majority of its time, and that's where it's going to hang out. It's going to get ready to kind of go through mitosis. And then in our M phase or mitosis phase, this is when the cell is going to separate out. So again, majority of it spends in interphase, and then we also have our M phase, which is the actual division portions. Now, we have two gap phases, G1 phase and G2. G1, remember, it's growing larger, it's making copies of its organelles. S phase, synthesizing. Remember, DNA synthesize, making copy of DNA. And then G2, again, it's going to go ahead and grow more, make those proteins, and then get all the things or reorganize this content in order to kind of get ready for that cell division phase. Then again, M phase is where it's actually going to go ahead and separate out. So as we're going through these phases, each portion is going to have a checkpoint because, again, it's vitally important to make sure that everything is going as planned so we don't have any issues down the line. So we do have 3 checkpoints. And just like the previous slide where we had those gap phases and synthesis phases, you can see that we have those checkpoint phases. So we have our G1 checkpoint, our G2 checkpoint, and then we have our M phase checkpoint here. So in our G1 checkpoint, all cells check whether internal and external conditions are right for cell division. Again, we got to make sure that we're in the correct conditions so we can actually go through that cellular division. In G2 checkpoint phase, the cell is going to check if the DNA is free of damage and whether the DNA was completely copied during S phase. Remember that S phase synthesis, we're creating that second copy so that when we do separate out and we do divide, they both have those pieces. So we want to make sure that DNA is absolutely correct. And then finally, right before we go through mitosis, cell is going to check whether sister chromatids are correctly attached to the mitotic spindles. So again, so it can actually pull apart and distribute that DNA from that original piece to those dotted portions. So again, we have these 3 checkpoints to make sure that everything is going exactly as planned. So then when we have our original piece, we are going to end up with 2 identical daughter cells. All right, and that is our kind of quick overview as to what our cell cycle is. There are going to be other lectures to talk about mitosis and meiosis to kind of really understand exactly what's going on with those individual phases. But again, this one was just a quick introduction as to what the cell cycle is. So I have 3 questions for you. I'm going to go ahead and give everybody about 5 seconds to pause this and see if you can answer these. All right, welcome back. And this is what you should have gotten. So chromosomes are duplicated during what phase of the cell cycle? It should have been S. Remember, S stands for synthesis. So we are creating that copy. Remember, G1 phase, they're growing. G2, they're getting ready for that division, settling everything out. And then our M phase is that actual division portion. So in this case, S synthesis, synthesize is making. So in this case, we are making our chromosomes or duplicating them in this phase. During division of cells, DNA into 2 identical sets of chromosomes occur during what phase? Again, M phase, division, dividing out. M phase, we're going to be going from R1 to R2. This is the only phase we're physically separating them out. What is necessary for a cell to pass the G2 checkpoint? We got to make sure we have accurate and complete data. Or I'm sorry, not data. We need to make sure we have accurate and complete DNA replication. So we want to make sure that our DNA looks exactly the same. So when we do divide that out, this is getting the exact same pieces and this is getting the exact same pieces. All right, and that is going to conclude the lecture. So as always, if you do have any questions, comments, or concerns, please feel free to reach out, and I look forward to seeing everybody in the next lecture.